Hello friends, welcome to a lecture on neuron structure and function of neuron, condition of nerve impulse in a neuron, neurotransmitters, types and functions. The main objectives of today's lecture are to have a precise discussion about neurons, to discuss structure and function of neuron, to help students understand the condition of nerve impulse in a neuron, to have precise discussion about neurotransmitters, and to understand precisely types and functions of neurotransmitters. Let us first of all discuss neurons. Neurons are cells of the nervous system that transmit signals throughout the body. Neurons can do many different things, from sensing external and internal stimuli to processing information and also directing muscle actions. Neurons are the highly specialized cells of all nervous systems. For example, central nervous system and peripheral nervous system that are tasked with transmitting signals from one location to another. These cells accomplish this role through specialized membrane-to-membrane junctions called synapses. Most neurons possess an axon, which is long protrusion from the body soma of the neuron to the synapse. Axons can extend to distant parts of the body and make thousands of synaptic contacts, such as is the case with the central nervous system neurons of the spinal cord. Axons frequently travel through the body in bundles called nerves. The synapses are termed presynaptic and postsynaptic. The presynaptic synapse will release secretory granule contents in response to the propagation of an electrochemical signal down its axon. The released substance, termed a neurotransmitter, will then most likely bind to a specific receptor on the membrane of the postsynaptic synapse, thereby propagating the initial signal to the next neuron. The human nervous system is composed of hundreds of different types of neurons. These include sensory neurons that transmit physical stimuli such as light and sound into neural signals, and motor neurons that are responsible for converting neural signals into activation of muscles or glands. Moving on, let us discuss classes of neurons. Based on their roles, the neurons found in the human nervous system can be divided into three classes: sensory neurons, motor neurons, and interneurons. Now, sensory neurons. Sensory neurons get information about what is going on inside and outside of the body and bring that information into the central nervous system so it can be processed. For instance, if we picked up a hot coal in hand, sensory neurons with endings in our fingertips would convey the information to our central nervous system (CNS) that it was really hot. Now, motor neurons. Motor neurons get information from other neurons and convey commands to our muscles, organs, and glands. For instance, if we picked up a hot coal, the motor neurons innervating the muscles in our fingers would cause our hand to let go. Now, interneurons. Interneurons, which are found only in the central nervous system, connect one neuron to another. They receive information from other neurons, either sensory neurons or interneurons, and transmit information to other neurons, either motor neurons or interneurons. For instance, if we picked up a hot coal, the signal from the sensory neurons in our fingertips would travel to interneurons in our spinal cord. Let's now discuss classification of neurons on the basis of shape. The most unique and important structures of a neuron are the long extensions that extend out from the cell body. There are two types of extensions: dendrites and axons. Dendrites are extensions of neurons that receive signals and conduct them towards the cell body. Axons are extensions of neurons that conduct signals away from the cell body to other cells. Both axons and dendrites can extend far away from the cell body, and some human axons can reach lengths of over 3 feet. But that's nothing compared to giraffes, which have axons as long as 15 feet. Neurons can be classified according to the number of processes that extend from the cell body as multipolar neurons, bipolar neurons, and unipolar neurons. Multipolar neurons are the most common type. They have several dendrites and one axon extending from the cell body. Bipolar neurons have two processes extending from the cell body, an axon and a single dendrite. This type of neuron can be found in the retina. Unipolar neurons are generally sensory, that is afferent neurons that have a single process, which then divides into two. One of the two processes extends outward to receive sensory information from various areas of the body, while the other process relays sensory information towards the spinal cord or brain. Now let us discuss the basic functions of a neuron. Neurons have three basic functions. These are to receive signals or information, integrate incoming signals, that is to determine whether or not the information should be passed along, and communicate signals to target cells, that is other neurons or muscles or glands. These neuronal functions are reflected in the anatomy of the neuron. Now, electrical signals in neurons. All living cells have a separation of charge across the cell membrane. This separation of charge gives rise to the resting membrane potential. Neurons and muscle cells both use brief changes in this resting membrane potential to quickly send signals from one end of the cell to the other. In neurons, electrical signals called action potentials propagate from the cell body down the axon to the synaptic terminals where stored neurotransmitter is released. Action potentials are transient. All or none changes in resting membrane potential that travel along the axon at rates of 1 to 100 meters per second. Myelin, a fatty insulating material derived from the cell membrane of glial cells, covers the axon of many vertebrate neurons and speeds the conduction of action potentials. The importance of this myelin covering to normal nervous system function is made painfully obvious in individuals with demyelinating disease in which the myelin covering of the axons is destroyed. Among these diseases is multiple sclerosis, a demyelinating disease of the central nervous system that can have devastating consequences including visual, sensory, and motor disturbances. Although neurons share many of the features found in other cell types, they have some special characteristics. For example, neurons have a very high metabolic rate and must have a constant supply of oxygen and glucose to survive. Also, mature neurons lose the ability to divide by mitosis. Until the late 20th century, it was thought that no new neurons were produced in the adult human brain. However, there is evidence that, at least in some brain areas, new neurons are produced in adulthood. This finding suggests an exciting avenue for possible approach to treating such common neurological diseases such as Parkinson's disease and Alzheimer's disease, which are characterized by the loss of neurons in certain brain areas. Let us now discuss neurotransmitters. Neurotransmitters are endogenous substances that act as chemical messengers by transmitting signals from a neuron to a target cell across a synapse. Prior to their release into the synaptic cleft, neurotransmitters are stored in secretory vesicles called synaptic vesicles near the plasma membrane of the axon terminal. The release of the neurotransmitter occurs most often in response to the arrival of an action potential at the synapse. When released, the neurotransmitter crosses the synaptic gap and binds to specific receptors in the membrane of the postsynaptic neuron or cell. Neurotransmitters are generally classified into two main categories related to their overall activity: excitatory or inhibitory. Excitatory neurotransmitters exert excitatory effect on the neuron, thereby increasing the likelihood that the neuron will fire an action potential. Major excitatory neurotransmitters include glutamate, epinephrine, and norepinephrine. Inhibitory neurotransmitters exert inhibitory effects on the neuron, thereby decreasing the likelihood that the neuron will fire an action potential. Major inhibitory neurotransmitters include GABA, that is gamma-aminobutyric acid, glycine, and serotonin. Some neurotransmitters can exert both excitatory and inhibitory effects depending upon the type of receptors that are present. In addition to excitation or inhibition, neurotransmitters can be broadly categorized into two groups, defined as small molecule neurotransmitters or peptide neurotransmitters. Many peptides that exhibit neurotransmitter activity also possess hormonal activity, since some cells that produce the peptide secrete it into the blood, where it then can act on distinct cells. Moving on to classification of neurotransmitters. Neurotransmitters fall into several chemical classes based on the molecular structure. The major types of neurotransmitters include acetylcholine, biogenic amines, and amino acids. The neurotransmitters can also be classified based on function, that is excitatory or inhibitory, and action, that is direct or neuromodulatory. Now let us discuss acetylcholine as a neurotransmitter. Acetylcholine, ACH, is a simple molecule synthesized from choline and acetyl coenzyme A through the action of choline acetyl transferase. Once released, acetylcholine binds to postsynaptic receptors and is degraded by acetylcholine esterase. Acetylcholine releasing neurons are also found in the central nervous system, CNS. Neurons that synthesize and release ACH are termed cholinergic neurons. When an action potential reaches the terminal button of a presynaptic neuron, a voltage-gated calcium channel is opened. The influx of calcium ions, calcium 2 positive, stimulates the exocytosis of presynaptic vesicles containing ACH, which is thereby released into the synaptic cleft. Once released, acetylcholine must be removed rapidly in order to allow repolarization to take place. This step, hydrolysis, is carried out by the enzyme acetylcholine esterase. The acetylcholine esterase found at nerve endings is anchored to the plasma membrane through a glycolipid. Acetylcholine receptors are ligand-gated cation channels composed of 4 different polypeptide subunits. The activation of acetylcholine receptors by the binding of acetylcholine leads to an influx of sodium Na positive into the cell and the efflux of potassium K positive, resulting in a depolarization of the postsynaptic neuron and the initiation of a new action potential. Now let us discuss biogenic amines as neurotransmitters. Biogenic amines include the catecholamines such as dopamine, norepinephrine, and epinephrine, as well as indole amines such as serotonin and histamine. Dopamine and norepinephrine are synthesized from amino acid tyrosine. Serotonin is synthesized from tryptophan. Histamine is synthesized from amino acid histidine. Biogenic amines are distributed in the brain, where they play a role in emotional behavior and help in regulating the biological clock. Additionally, some motor neurons of the autonomous nervous system release catecholamines like norepinephrine. Norepinephrine, dopamine, and histamine can be excitatory or inhibitory depending on the receptor type. Addictive drugs such as cocaine and amphetamine exert their effects primarily on the dopamine system, while addictive opiates and functional analogues of the opioid peptides which regulate dopamine levels. Now we have amino acids as neurotransmitters. Glutamate and gamma-aminobutyric acid, that is GABA, are amino acid based neurotransmitters. The most prevalent transmitter in the human brain is glutamate, which promotes excitatory effects by increasing the probability that the target cell will fire an action potential. The next most prevalent is GABA, which is inhibitory at more than 90% of the synapses that do not use glutamate. Neuropeptides such as substance P and endorphins are strings of amino acids that are important in the mediation of pain signals. Enkephalin activity increases dramatically in pregnant women in labor. Endorphins are released in the so-called runner's high. There is also evidence that neuropeptides such as gut-brain peptides are produced by non-neural tissues in the gastrointestinal tract. Next important neurotransmitter is GABA. Several amino acids have distinct excitatory or inhibitory effects upon the nervous system. The amino acid derivative gamma-aminobutyric, also called 4-aminobutyrate, that is GABA, is a well-known inhibitor of presynaptic transmission in the central nervous system and also in the retina. The formation of GABA occurs by the decarboxylation of glutamate catalyzed by the glutamate decarboxylase, that is GAD. GAD is present in many nerve endings of the brain as well as in the beta cells of the pancreas. Neurons that secrete GABA are termed GABAergic. GABA exerts its effects by binding to two distinct receptors, GABA A and GABA B. The GABA A receptors form a chlorine channel. The binding of GABA to GABA A receptors increases the chlorine conductance of presynaptic neuron. The anxiolytic drugs of the benzodiazepine family exert their soothing effects by potentiating the responses of GABA A receptor to GABA binding. The GABA B receptors are coupled to the intracellular G protein and act by increasing conductance of an associated potassium positive channel. Next important neurotransmitter is catecholamines. The principal catecholamines are norepinephrine, epinephrine and dopamine. These compounds are formed from phenylalanine and tyrosine. Tyrosine is produced in the liver from phenylalanine through the action of phenylalanine hydroxylase. The tyrosine is then transported to catecholamine secreting neurons where a series of reactions convert it to dopamine, to norepinephrine and finally to epinephrine. Catecholamines exhibit peripheral nervous system excitatory and inhibitory effects as well as actions in the central nervous system such as respiratory stimulation and an increase in psychomotor activity. The excitatory effects are exerted upon smooth muscle cells of the vessels that supply blood to the skin and mucous membranes. Cardiac function is also subject to excitatory effects, which leads to an increase in heart rate and in the force of contraction. Inhibitory effects, by contrast, are exerted upon smooth muscle cells in the wall of the gut, the bronchial tree of the lungs, and vessels that supply blood to skeletal muscles. In addition to their effects as neurotransmitters, norepinephrine and epinephrine can influence the rate of metabolism. The catecholamines bind to two different classes of receptors termed the A and B adrenergic receptors. The catecholamines therefore are also known as adrenergic neurotransmitters. Neurons that secrete them are adrenergic neurons. Norepinephrine secreting neurons are noradrenergic. The adrenergic receptors are classical serpentine receptors that couple to intracellular G proteins. Some of the norepinephrine released from presynaptic noradrenergic neurons recycled in the presynaptic neuron by a reuptake mechanism. Epinephrine and norepinephrine are catabolized to inactive compounds through the sequential actions of catecholamine O-methyltransferase, that is COMT, and monoamine oxidase, that is MAO. Compounds that inhibit the action of MAO have been shown to have beneficial effects in the treatment of clinical depression, even when the tricyclic antidepressants are ineffective. The utility of MAO inhibitors was discovered serendipitously when patients treated for tuberculosis with isoniazid showed signs of an improvement in mood. Isoniazid was subsequently found to work by inhibiting MAO. With that, we come to the end of today's lecture. Hope you have understood it well. Thank you and goodbye.